Okay, thank you so much. I would really like to thank the organizers for giving me this opportunity to share our work with all of you from wherever you are linking in. Okay, so everybody is very familiar with light sensing in nature. So it's used by humans, of course, to see, by animals, plants, algae, bacteria, for processes like vision, regulation of signaling, transcription, and all of these processes are mediated by a plethora of light-sensing molecules. And only photoreceptors are being studied for many decades, but only recently it has come more to the attention of neuroscientists due to the organism that form this nice green film on ponds. This organism is Chlamydomonas, an algae. And this algae has an eye spot, and these are, I don't know how well you can see it, flagella. So when the sun hits the pond, or in this case, in the lake, then the light pushes the algae to one side due to the flagella moving, and the light hits the eye spot. And it was only in 2003 that Georg Nagel, Peter Hegemann and friends identified the actual molecule mediating this, and that's channelrhodopsin. This was had never a rhodopsin which had never seen before, because here the chromophore is not embedded and functions through a G protein coupled receptor, but through actually a channel itself. So the light directly allows to open this channel. And this, of course, was picked up immediately by neuroscientists, as it allows channel opening in neurons, just like in their endogenous situation, with millisecond time scales. And of course, in vivo, you can also do this. Again, light fibers get inserted in the brain, and genetically targeted channelrhodopsins then allow you to activate or silence neurons. Enough said here. What's a little bit less known is, against the photo sensors, there's also light emission in nature, and actually quite a lot. Probably most of you have seen fireflies in the summer. Another group of light-emitting organisms are these dinoflagellates, which make the nice blue waves. And this biological light, or bioluminescence, so light emitted by living organism, can be found not only in fireflies and dinoflagellates, but in a large conglomerate of bacteria, fungi, and in fact, the vast majority of bioluminescent organisms are living in the ocean. On the other hand, 75% of ocean dwellers are actually bioluminescent, which kind of makes sense, because how else are you going to regulate traffic at the ocean floor where there's no sunlight? So how does biological light get generated? It requires an enzyme. So for the firefly, it will be an insect, the firefly luciferase, and it requires a substrate, so a small chemical molecule called a luciferin, and the luciferin gets oxidized, and this generates an excited state, which then produces a photon. For the firefly, this process needs not just oxygen, but also ATP. Now, for the marine organism, they don't use the D-luciferin the firefly uses. They use, a lot of them use celanterazine, which has a different chemical structure. And this conversion, this oxygenation and light production does not require ATP. So marine luciferases, in that sense, have the advantage. There's no siphoning of ATP while the light gets produced. To just give you a feel for how much light is produced, this is Gaussian princeps. a copepod. It expresses a luciferase called Gaussian luciferase. It uses solanterazine, and it produces 4.2 times 10 to the 24 photons per second per mole. So the light generation is quite considerable. Now, in nature, we have mouse. Okay. Hold on, my mouse is cooperating. There. In nature, bioluminescence is used for a variety of processes, and all sorts of vital activities can be involved. For example, bioluminescence is used to hunt prey. Anybody who watched the movie Finding Nemo will remember that from the lanternfish. It's used by other organisms to defend against predators. It's used to find mates. And in this table here from Steve Haddock's review kind of nicely lists all the different applications. The pictures here show this a school of flashlight fish. And I don't know how well you can see it, but this is white light. You see the whole group. When you turn the white light off, you see the little blinking signals from the bioluminescence. That's how these fish, once they find a food source, they all conglomerate on it. And here you can see kind of the overlay. This is work from Stefan Herlitz's lab. So in the lab, you might have come across bioluminescence in two instances. It's most used in vitro with a luminometer, so a plate reader. It's used for reporting transcription, for reporting molecular interaction, etc. In vivo, you might have come across it through the in vivo imaging system, or IVIS. This is a system where you don't have to kill the animal to monitor, for example, tumor cells. You just anesthetize it, inject the luciferin, image it, and you can do that throughout the lifetime of an animal. So, but none of these are really what we do. What we want to use bioluminescence for, and have been using it for, is to activate, to drive photosensing molecules. And we call this BioLuminescent OptoGenetics, or BLOG. So what's in it when you combine a light sensor with a light emitter? What specifically does this enable? And there are three key aspects to this where BLOG can specifically enable something other approaches cannot necessarily do, that I want to highlight during the next 30 minutes. The first one is bimodal optochemogenetic activation. That's when you combine a luciferase with an opsin. The second is an all-molecular feedback system, when you combine the opsin with a luciferase that is dependent on an intracellular mediator. And the third one is the optical synapse, when you combine a presynaptic light emitter with a postsynaptic light sensor. So let's go to the first item, the bimodal opto and chemogenetic control of the same cell. This started with luminopsins. So luminopsin, or luminescent channelrhodopsins, it's basically a channelrhodopsin where we tether to the N-terminus a luciferase. And the EYFP or YFP, the fluorescence reporter, is just to label the cells. So when the luciferase gets its substrate, it emits photons, and these photons get captured by the chromophore and open the channel. So to show you kind of the history of this, the first one was LMO1 with just wild-type Gaussian luciferase and the standard channelrhodopsin 2. And this is in HEK cells. The blue line is the bioluminescence emission, and the black line is the invert current in HEK cells. And you can see just the tiniest little inflection here. So the next, this we didn't even put in neurons because it was just not big enough. So then we thought, well, maybe we take an opsin that has higher light sensitivity. So we used VCHR1, the Volvox channel, again with wild-type luciferase in LMO2. And now you can see the inward current is just a little bit bigger. So we were encouraged to put it in neurons, and in neurons, we could show that it does change the threshold, in that when you inject current and there is no CTZ, no luciferin present, you get your spikes. If you add the luciferin and the bioluminescence opens the channel, you get more spikes. But then only with the third version, LMO3, we actually could drive action potentials. And what we did here is we still use the Volvox channelrhodopsin, but this time with a luciferase, which has like 10 times more light emission. So now the inward current is pretty big, you get action potential firing, and we actually have done a number of studies using this tool, and I will show a few examples of those. So by now, we are at LMO7, and if you look at the LMO3 in this graph here, that's the black line. So the LMO7 is still has a lot higher inward current here in HEC cells. LMO7 it's still VCHR1, but the light emitter now is a construct from Nathan Shainer's lab, and it's a fusion of M-neon green, the fluorescent protein, with ECAS, which is a mutated version of the oplophorus luciferase, and this basically Bragg probe then activates the channel. You can see here the inward current amplitude of the size of the current. This graph here shows you the coupling efficiency. Coupling efficiency is basically if you plot the photocurrent you get with LED versus the photocurrent you get with CTZ. You can see with the LMO7 that you can get almost as much inward current as with LED, with the bioluminescence light, as with LED. So these examples here, you can see that it's always in these three examples the same opsin, but luciferases with increased light emission. Here I'm showing you an example of the same luciferase, SPG look, so Gaussian variant, but with two different opsins, VCHR1 and here Sharif. And the LED photocurrent, it's a huge inward current, and that also in this case is reflected with the bioluminescence. You get an equally big step down here. So we are continuously making new luminopsins, and I'm not going to go into any details, just want to highlight here. So we match the light emission of the luciferase to the peak light activation of the opsin. For instance, the more redshifted with red luciferases. The arrangement of our constructs, most of them have the luciferase outside in the extracellular space, but the whole series of red shifted ones, actually we have the luciferase inside. The chromophore of the opsin doesn't care whether the light comes from top or bottom. Of course, we are always following the Optogenetics crowd for their most recent, new, more sensitive opsin. So we are trying to trying out the ones with the real high light sensitivity, and of course from our side then with light emitters that give the highest light emission. I just want to point out that there is no straight correlation. So if you take the highest light emitter with the highest light sensitive opsin, that doesn't automatically make a good luminopsin, because what matters really is that part of the activation of the opsin is mediated by first resonance energy transfer, and so the energy donor and energy recipient have to be matched in order to get a good result. And that just comes down to trying out all these different constructs. All right, so now I want to show briefly 3 applications where we actually have used luminopsin 3. 2 of them are in disease models, and one is a little bit more systems neuroscience applied. So for spinal cord injury, we used LMO3. This is a rat model of spinal cord injury, a contusion injury, so not a complete severance of the cord, but a contusion. So usually the neurons below the contusion are kind of numbed and they don't engage anymore. So we figured if we put LMO3 in here and the neurons express LMO3, we can activate them and kind of re-engage them into transmitting communication. The kind of overall experimental design here is, first we had a surgery where it's an experimental model, so we first put the LMO3, the AAV, into the spinal cord so that it can express. At this point, we also implanted a lateral ventricle cannula for easy application of the luciferin. Then we would do IVIS imaging to see that the LMO is expressed properly and in the right spot. And then we do the contusion surgery. So day after the surgery, we start the CTZ injections every other day for 2 weeks. And then the behavioral testing to see whether there's a better recovery. So we did this with synapsin-driven LMO3 and with HB9-driven LMO3. Synapsin is generic neuron promoter, and HB9 is a little bit more motor neuron focused. So the IVIS imaging here, one example is shown the distribution exactly at the part of the spinal cord where you expect to find it. It also shows you the timeline. So we do this basically every other day for 2 weeks. So all the neurons which are situated below the site of the injury get an activation that peaks at about 15 minutes and then slowly tapers off. And this was also nicely matched by the changes in neural firing rates when we recorded from in the spinal cord from the LMO3 expressing neurons. In the spinal cord field, to assess the effect of a therapy, what people do is they let the animal walk and apply what's called the BBB score. So BBB score of 20 means the animal walks on all 4 limbs. There's no impairment anywhere. So all the animals showed this. Then a day after the injury, all the animals do not move, which is also expected. But then just spontaneously over time, there's a certain degree of recovery after the contusion injury. But the recovery is kind of all in this gray box here. And the gray box is anything below a BBB score of 10. Anything over 10 means the animal is actually weight supporting on all 4 limbs. So the spontaneous recovery does not get to that stage. However, with this, the bioluminescence light activation of opsins below the contusion injury, either with the synapsin promoter or the motor neuron promoter, led to significantly improved motor recovery in this system. Now, more recently, we actually, I showed you earlier the comparison of the SBG loop with VCHR1 versus Sharif. So with Sharif being that much more light sensitive and giving a much larger response to the same amount of light, we now went ahead and did this basically the same experiment, but with the synapsin promoter driven with intraperitoneal injection of the luciferin, and that similarly led to a robust increase in motor recovery in these animals. So that's one of the advantages to working towards ever more potent luminopsins, that you can then go to something like intraperitoneal injection to get the same effect. The second example was a mouse model of Parkinson's disease, the PITX3AK mice. AK is for aphakia. the eyes of these animals don't have any lenses. They have a bunch of other problems, but for us here, the main thing is that they fail to develop dopaminergic neurons in the substantia nigra, and thus they show the typical sensory motor deficits specific to deficits in the nigrostriatal pathway. So in the rotor rod, the heterozygotes or the wild types, they hang on usually on average over 60 minutes, seconds over a minute. And then the PIDX3 homozygous mice, they don't have that motor coordination. They fall off about under 30 seconds. So what we wanted to do here is transplant neural precursor cells that express luminopsin 3 bilaterally in the striatum of these animals, and then see if we can, over time, improve their motor performance. So we already had embryonic stem cells that express LMO3, so we derived neural stem cells from these ES cells and then implanted those bilaterally into the striatum of the PIDX3 mice. And this is, again, an IVIS image through the closed skull of these mice. The experiment then was done. They would be trained on the rotor rod, and the rotor rod pre-experimental testing was scored. Then we would do the cell implantation, and after a week, 10 days of daily CTZ or vehicle application. So for 10 days, we would basically stimulate the neural precursor cells sitting in the striatum. And then afterwards, we had the post-treatment rotor rod testing. So if the animals had the cells, so the LMO expressing neuronal precursor cells and the vehicle for these 10 days, there was no improvement. If the animals had just CTZ without the cells, there was also no improvement. But if they had the LMO3 expressing neural precursor cells in the striatum and received the 10 days of stimulation, there was a robust improvement in motor recovery. So my third example, it's not a preclinical model. This is an application of BLOG where we really took advantage of the unique aspect of bimodal activation. So the channelrhodopsin doesn't care that there's a luciferase hanging off it. So you can stimulate it with LED or lasers in just the same way as you would do for a standalone opsin, so classical optogenetic interrogation. But at the same time, you can also apply chemogenetic drive to the very same molecule and the very same cell that expresses this molecule. So we applied this in one of our areas of interest, which is to study the effects of early postnatal neuronal excitation on the kind of layout of neural circuits and the consequences in adult behavior and underlying neural circuitry. So what we did here is we made a conditional knock-in mouse expressing LMO3. So this animal was then crossed with different Cree driver lines. The pups of these crosses were then stimulated or intraperitoneally injected once a day, postnatal day 4 to 14, which is, of course, very convenient to light up the brain just by an intraperitoneal injection. So every day, day 4 to 14, they would kind of have this peak expression of light. So their neurons would be firing once a day and basically being hyper excited. Then after the 14 days, the animals would just grow up normally, be adults. And when they were adults, we would send them through a whole array of behavioral tests, but also then use fiber optics for precise interrogation. So the Cree drivers we used were, probed the dopaminergic system. DLX56 is frontal cortex, interneurons. EMX1 is basically pyramidal cortical neurons, and then pavalbumin is interneurons in the cortex. So these 4 strains were sent through a whole battery of behavioral tests, and what we found, the most striking results and results we then followed up on were with the EMX1 LMO3 mice. So animals that were hyperexcited in all pyramidal neurons in the cortex had a showed decreased social approach. So their social interaction was somewhat impaired. These same mice also showed increased grooming or increased repetitive behavior. Now, these 2 items are key features of autism spectrum disorders. So then we took the adult animals and we wanted to see how the neuronal firing underlying these 2 behaviors is changed. So this is a schematic of the experimental setup. Laminar probes were inserted in the prelimbic area of the medial prefrontal cortex. This is in anesthetized animals, we would record baseline, then 10 seconds every 60 seconds of light pulses for 10 minutes, and then we would continue to record. So what we found is overall for pyramidal neurons, a decreased baseline firing and enhanced direct excitability. So the firing rate of pyramidal neurons in hyperexcited animals was always lower, basically in the basic state, but then they overshot, as you can see here, when they were excited by light pulses. And that's shown in the graphs here. And for the interneurons, we found actually that they were also significantly reduced. Their responses were reduced at baseline in the hyperexcited animals, but different from the pyramidal cells, they were also recruited less effectively by light stimulation. So this change in excitation inhibition balance, you would expect to also see effects on neural oscillations. And what you would expect is a shift to higher power, especially in the lower gamma range. And that's what we actually see here. So during light stimulus, the CTZ treated animals, so the pink line, the developmentally hyperexcited animals show significantly greater power in both beta and gamma, indicating that they may have experienced altered excitation inhibition balance. So in summary, developmental pyramidal hyperexcitation leads to decreased baseline firing and enhanced stimulus-evoked firing and increased power in beta and gamma range. And we are following these up with recordings from different areas in the brain and different targeting of sublayers in the cortex. In the context of this presentation here, the bimodal opto and chemogenetic control of the same cell, here block is unique in that you can really activate entire neuronal populations and then later on interrogate the same neurons that got manipulated here at a very precise spatial and temporal scale. So moving to the next item on my list, this is the aspect that having a light emitter that's genetically encoded, the light emission itself can be made dependent on intracellular processes. So the example I'm showing you here is, here it's made dependent on calcium. So calcium-dependent luciferase, which we call luminescence, are basically, in this case, a split Gaussian luciferase, the SPG look, and it's split by a CAM-M13, calmodulin M13 calcium sensing unit, just like all the GCAMs are. So in the presence of CTZ alone, you shouldn't see bioluminescence because the molecule is split apart. In the presence of calcium, the molecule gets the two halves get reunited. But in order to get light emission, you always need both. You need calcium to reunite it, and you need the CTZ to actually get the light emission. So this is shown here in this graph. What we used here were HeLa cells. And in HeLa cells, if you add histamine to HeLa cells, internal calcium stores are dumped out, and you should see a calcium response. And that's what you see here. These are two versions of the luminescent 4 and 7. They have different CAM-M13 moieties. The one in LMC4 is from GCAM6F. So what you see is when you add the CTZ initially, you do see a background, and different bioluminescent calcium channels have different degrees of background. And then with the histamine, so with the dumping of the internal calcium stores, the response, the bioluminescence light goes up. What's important here is, because you can use this for imaging, there are plenty of bioluminescent calcium imaging constructs on the market. What's important for us is we want to use this to drive opsins. So the background light emission with just CTZ and not just background calcium shouldn't be high. It should be under the level required for driving an opsin, while once calcium comes in, the light emission should be able to drive an opsin. So what really matters here is the delta between the CTZ and the histamine, which is actually excellent for the luminescent 4. And this preferred kinetic was also shown in vivo experiment by Manuel Gomez Ramirez in Chris Moore's lab. So here they transduced cortical neurons with luminescent 4. When you just add CTZ, which was applied here through the lateral ventricle, that by itself, CTZ by itself, you don't get much of a response. Once you inject NMDA, which then makes the neurons fire, you see an increase in light emission just in the neurons that express the luminescent, but not in neurons that don't. So we wanted to now directly test if we can actually drive an opsin in neurons that express both an opsin and a luminescent. So we assessed by whole cell patch clamp whether BioLight, activity-dependent BioLight, can influence single neuron activity. So here you see the luminescent expression. It has a P2AD tomato, so it's red. And then the opsin expression in the same neuron. In this case, the overlay, and then these neurons got patched. Now, the experiment was basically taking advantage of a classical input-output curve. So a neuron which expresses an opsin and a luminescent, if it gets injected with current, you should see increased spiking with increased current, and you get this typical relationship in the input-output curve. So what we wanted to know is if we then add CTZ, so BioLight gets emitted, and the opsin is an excitatory opsin, we should see a left shift in this curve, because the threshold is actually is lifted up, so that we should see a higher count of spikes at an earlier time point. And then the opposite trend should occur if the opsin is an inhibitory opsin. So as calcium comes in, CTZ is present, light goes on, and now we have, let's say, chloride coming in. we should see a dampening of the response. So that's shown here. Again, this is the schematic when there's no opsin in there and we give CTZ, you just get light emission. It should be just the control, and that's exactly what it looks like. So before and CTZ completely overlap. If there's an excitatory opsin, then, as I said, we should see the left shift. And again, that's what we see here when the neurons, when we record from the neuron. So this is always the same neuron with before CTZ, it gets a sweep, and after CTZ. And then with the inhibitory opsin, again, we expect the right shift. And again, that's what we see. So we also wanted to assess this for a network of neural activity. So we use multi-electrode arrays and plated neurons that expressed, in this case, ACR2, so an union channel. So we should see an inhibitory effect, presence of calcium, presence of CTZ turns the light on, and we should see increased inhibition. And that is what we see. So this is the LED control. You see the inhibition and firing, and then with CTZ addition, you get inhibition after the CTZ for a considerable amount of time. And you don't see this if you apply the CTZ, but there is no luminopsin in here. Okay, so as I said, the light emission can be made dependent on intracellular processes, in this case, calcium. However, you can also use other intracellular messengers, like such as CAMP. What you can use the system for is a true molecular integration. Let's say, envision an epileptic focus, and the cells express an inhibitory opsin and a calcium sensor, a calcium sensing dependent luciferase. As the epileptic process starts, calcium comes in, light goes on, and the cell itself basically dampens its activity. So that's the type of molecular integration we envision here. So the third aspect, and very exciting aspect, is since we have a light emitter that's genetically encoded, it can be expressed in a different cell than the light sensors. So in this case, photons can be used for cell-cell communication. So what I showed you thus far is an application of intraluminescence. So the opsin and the light emitter, the luciferase, either calcium dependent or not, are within the same cell, either in the cell or tethered. If you break it up so that the sender cell has a luciferase and the receiver cell has an opsin, you can use the photons exchanged here as a communication device. And if you do this in neurons, you can put the luciferase in a presynaptic neuron and the opsin in a postsynaptic neuron. And in this instantiation, the luciferase is actually targeted to vesicles, so that the release of the luciferase is dependent on the presynaptic neuron to be activated. So the neuron is active, it dumps out the luciferase, just like it dumps out neurotransmitters. In the presence of CTZ, the luciferin light gets emitted, opens the opsins. And we showed this in MEA experiments. So this is just blue light, so all the opsins are directly opened. This is with CTZ, and what you see whited out here is the artifact when you add liquid to an MEA. But what you see is an increase in firing due to the luciferase released in the synaptic cleft and then opening the channels in the presence of CTZ, and this does not happen with vehicles, so in the absence of light, biolight. And the same thing is true for inhibition, again, just directly blue light. If you use the CTZ, then you get inhibition, and you don't get the inhibition with the vehicle. This shows you a raster plot to kind of see all the electrodes in an MEA. Blue light here activates, and then the CTZ addition activates very robustly. Blue light inhibits, and again the CTZ very robustly inhibits the postsynaptic neurons. We also showed that the interluminescence is dependent on the presynaptic vesicle release. If you add botulinum toxin to cultures, botulinum toxin inhibits the fusion of vesicles and the release of vesicle content. So here we add neurons are firing, we add synaptic blockers, and then an electrical or light stimulus here activates the presynaptic neuron. In the presence of CTZ, the light emission activates the postsynaptic neuron. Once you add the botulinum toxin, all of that doesn't work anymore because there's no release. But the neuron is still healthy. You can still directly fire it up with blue light, with LED. We also showed that the interluminescence mediated by bioluminescence activation of the opsin. And here we took advantage of the fact that we used a step function opsin. Step function opsins you can activate with blue light, but you can shut them off with green light, as is shown here. So you fire the neuron, it starts firing with the blue light, you emit the green light, and it immediately turns off. And this turning off also is seen when the channel is fired up by biolight. So indeed, it is through the opsin that interluminescence is mediated. A separate independent confirmation of that is we used what we call a DUD channel. So we introduced 2 additional mutations in this step function opsin that makes the channel unresponsive to blue light. So when we go now through the biolight activation, again, it does not work if the opsin isn't working. All right, of course, most importantly, how does this work in vivo? So these were experiments done by Jeremy Murphy in Chris Moore's lab at Brown. So we were wondering if we can change network dynamics that are created by long-range cell type-specific communication. So glutamatergic neurons in the thalamus synapse with neurons in the cortex, including direct synaptic input to fast spiking interneurons, pathalbumin interneurons. So the animals used here are pathalbumin Cre expressing, and we injected a Cre-dependent version of the step function opsin. So it's only expressed in pathalbumin neurons. And the thalamus received AAV injections of the luciferase, the vesicle-targeted luciferase. And here you can actually see the pathalbumin interneuron, and the red dots around it are the contacting points from the neurons which are from the thalamus. So 2 groups of animals were used. One group, all the animals expressed the luciferase. So you see bioluminescence in both cases, whether they have an opsin or not. Bioluminescence emission is pretty equal, but only in the animals which have the opsin, CTZ application resulted in robust broadband gamma oscillations with the presence of CTZ. And it's not seen, if you give CTZ, you get the bioluminescence, but not the gamma oscillations when there's no opsin. Furthermore, the interluminescence gamma increase is localized more superficially in the cortex. This is kind of along the outside to inside of the cortex. That makes sense because the CTZ was dropped on top of the cortex here. So a tool that permits direct experimental control of the efficacy and form of synaptic transmission between synaptic partners is highly desirable for a lot of applications. For example, you can enhance or suppress endogenous patterns. You can change the valence of communication. For example, a presynaptic neuron synapsing to a postsynaptic neuron, where the postsynaptic neuron normally is excitatory, you can now make the communication, turn it into inhibition. You can strengthen desirable and dampen undesirable intercellular communication under pathological conditions. So there's a lot to be done with this tool in systems neuroscience and also possibly in preclinical models. So overall, I hope I was able to shed some light on the unique capabilities of BLOG. There are numerous questions we are following up on regarding the underlying mechanism of BLOG, especially for the interluminescence. At the same time, we are taking advantage of these different features for ongoing basic and preclinical studies. And of course, none of this would be possible without the people. These are the people from my lab who actually make it all happen in the trenches. And I feel privileged to be able to coach this talented group of people. I'm also lucky that I'm not coaching alone. I'm a member of a coaching team with my friends at the West Coast and East Coast, Nathan Shaner at UCSD, and Chris Moore and Diane Lipscomb at Brown University. And my students, as you can see here with these arrows here, my students and postdocs have benefited immensely from cross-training, spending time at UCSD and at Brown. And I want to close this with a big thank you to our financial supporters. The Keck Foundation was critical in launching us. The National Institutes of Health through the BRAIN Initiative have been continuously supporting this work. The National Science Foundation has immensely helped by powering our bioluminescence hub, and please do go to the website. I also want to mention the Craig Nielsen Foundation that generously supported especially the spinal cord work. And thanks everybody for listening in, and I'm happy to take questions.
Well, Professor Hochgeschwender, many thanks for this nice talk. I wait for some more questions from the students because we currently just have one, but I have two questions myself that I would like to ask, because I saw the development of luminopsins from 2013, the first paper, Ken Berglund. So I think in this paper, there was the idea to separate the luciferase from the channelrhodopsin to increase the efficiency of the effect size. Did you continue this research, whether it would improve to separate luciferase and the opsins from each other, or didn't it make sense?
Actually, we kind of hinted at the optical synapse at that point when we separate them in two different neurons. Separation in terms of luminopsins and in terms of better driving the opsin, at that point in 2013, the optical synapse would not have worked because the luciferase wasn't bright enough and the opsin was not necessarily sensitive enough. So it took quite a while to get to light emitters that emit enough light to actually function when the two are not connected. So in terms of efficient luminopsins, we still tether the two, but the more light emission you get, the freer you are to separate them either within the cell or now even across cells.
Okay, many thanks. Another question that I have is, luminopsin is actually a cool tool for Parkinson, as you showed in your, let's say, experiment. But something that I have in mind from one of our reviewers that was complaining about the fiber optics, that it would also affect the visual perception of the animal if there's a light source inside the brain. Did you notice whether the luciferase activity would cause an impairment in animals in a visual task, or is there some research going on?
We haven't tested that specifically. So with BioLight or bioluminescence inside the brain, there's no light getting outside which the animal would see. So in that sense, I wouldn't anticipate any problems on that end. If it affects the visual system, it would have to, the opsin or the luminopsin would have to be in the visual cortex to do so. So I can't see it, but then we haven't specifically looked at it.
Okay, thanks. So I would like to go over to the students' questions, and we still have just one, which is from Julia Bandura. Why does the firing frequency seem to max out? An example, the relationship is nonlinear when the excitatory opsin is stimulated.
So is that in conjunction with the lumicamsin, the calcium?
Yeah, sorry, that's my question over here. Yeah, I was just looking at like the gain of firing over here. Yes, this one.
This one.
Yes, thank you.
Right. So, oh, you're wondering why it maxes out?
Yes, yes.
I'm not sure actually. It's, I mean, if you look at the straight control, at some point you will plateau here as well. I would say it depends on how you set up the current injections, how you perform the sweeps.
Good to know. Thank you so much. And I just had a quick follow-up question if you have time. Just I was wondering whether these neurons expressing these luminopsins are at all like electrophysiologically different from wild type.
We haven't seen that. Actually, we are conducting a study where we look at neurons expressing different chemogenetic actuators, and we are looking more carefully into whether there are any morphological, functional, or transcriptome changes in those neurons. So that's ongoing. But in terms of their immediate, but that's after long-term application. In terms of their immediate reactivity, we don't see a difference to a wild-type neuron.
That's very cool. Thank you so much.
Unless, I have to add that, unless you overexpress the luminopsin, as the same thing if you overexpress opsins to past a certain level, the neuron doesn't like it anymore. So this is all within a physiological range of expression. So expression that you see an effect, but that you don't kill the cell.
Yeah, that makes sense. Thank you so much.
So we have one question from, let's have a look, was Juan Jose, who wrote directly to Jenny. Is it clear if stimulation of neurons with light could provoke the eventual degeneration for long-term applications, for example?
Yes, that's exactly one of the aspects we are looking into now in terms of long-term application of different actuators. And one of the motivations for us to use BioLight here is that it is definitely milder in terms of bleaching. You don't get anything like bleaching. It's not a laser or an LED. You have the light directly next to the chromophore of the opsin. You activate the opsin, but beside that, there is no effect of the light, especially no negative effect of the light. Yeah, one of the motivations for using this tool is exactly that you don't want to use LED for long term.
Thank you. Then we have a question from Kansarika. There's plenty of alpha-synuclein accumulation in PD neurons. Are there any differences in using this technique in healthy neurons versus Parkinson neurons?
So the question goes at what if the luminopsin, or any probe for that matter, is expressed in unhealthy neurons? Is that right?
That's how I understand the question. Maybe Kansarika could, yes, it's the question.
That's a question. Yeah, that's actually a reason we use the transplanted neural precursor cells here. So with the idea of, in PID extremis anyway, they don't have dopaminergic neurons in the substantia nigra, so there wouldn't be any way to reconstitute that pathway. So that's why we specifically use the transplanted cells. It would be interesting actually to see and express luminopsins, drive them with LED, drive them with biolight, and see if one or the other is more or less harmful to an already damaged neuron. That would definitely be interesting.
Then we have a question from Chris Popple. Can presynaptic membrane-bound or intracellular calcium-sensitive luciferases activate postsynaptic channelrhodopsins, or does this currently require vesicular location and release to achieve sufficiently small distance between luciferase and channel?
Right. That's also a question right on target. What we are doing actually is, so the vesicle targeted is a version of activity-dependent communication through the luciferase and the bioluminescence. What we also do, and we have experiments running on that, we anchor the luciferase into the presynaptic membrane, that it basically sticks out permanently into the synaptic cleft. That makes the light emission exclusively dependent on when you apply the CTZ. It's not anymore dependent on the activity of the presynaptic neuron, and that works as well.
Okay, so we have one more final question, which is to study the presynaptic postsynaptic connections, can the LMSO be targeted to specific phenotype of presynaptic cells or neurons?
Right. So basically, both components, the light emitter, the light sensors, are genetically encoded. So you can target them with whatever genetic cell-specific tool you have on hand. So you could try out, in addition, you can then use the spatial limitations of, let's say, a targeted AAV injection. So let's say you want to test communication between ventral hippocampus and areas in the prefrontal cortex. You can target the opsin very specifically in infralimbic versus other areas, and you can target your presynaptic neurons with the right Cree driver animal, make it very neuron specific. Yeah, that's definitely the idea with that.
Okay, it seems like that this was our last question. So I would really like to thank one more time our Professor Ute Hochgeschwender for this nice talk and also the questions, and hope to see you one time at DSFN or NVG.
Okay, well, thanks everybody for listening in.